replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
During DNA replication, one of the two DNA strands, the leading strand, is replicated continuously, or all at once, in the 5' to 3' direction. The other strand, called the lagging strand, is replicated discontinuously, or in pieces, in the 3' to 5' direction. This is necessary because DNA polymerase, the enzyme that synthesizes a new DNA strand using a template strand, can only add nucleotides to the 3' end of a polynucleotide strand. Overall DNA replication is semi-discontinuous. To begin DNA replication, a helicase enzyme attaches to the double helix and unwinds the two strands, exposing them as templates for replication. The point at which the two strands are being separated is the replication fork. The enzyme primase attaches to each of the template DNA strands. Primase synthesizes a short RNA primer on each template strand. The primers are assembled in the 5' to 3' direction. After the primer reaches a certain length, primase drops off of the template strand. A DNA polymerase molecule attaches to each DNA template strand at the 3' ends of the RNA primers. Each DNA polymerase begins adding DNA nucleotides to extend the primer in the 5' to 3' direction. As the replication fork opens, the DNA polymerase on the leading strand template continues to synthesize a new DNA strand. However, the DNA polymerase on the lagging strand template, which can only synthesize a new DNA strand in the 5' to 3' direction, is moving away from the replication fork. Therefore, primase must again make an RNA primer on the lagging strand template, and DNA polymerase must continue making DNA beginning at the new primer. This results in a series of short fragments, known as Okazaki fragments, on the lagging strand template. The Okazaki fragments on the lagging strand template must be connected to form a complete DNA strand. To accomplish this, a different DNA polymerase recognizes and removes the RNA primers and replaces them with DNA. However, the polymerase cannot form the bond between the DNA nucleotide at the 3' end of one fragment and the nucleotide at the 5' end of the adjacent fragment, so there is a nick in the DNA strand. The enzyme DNA ligase is needed to form this bond and eliminate the nick. The process of continuous DNA synthesis on the leading strand template and discontinuous DNA synthesis on the lagging strand template continues until the replication fork reaches either the end of the chromosome or another replication fork moving toward it in the opposite direction.